Welcome. So, have you ever looked at one of those big data-heavy questions on a biology paper and just thought, where do I even begin? Well, if you have, you are in exactly the right place. In this explainer, we're going to take a real AQA GCSE question and completely tear it apart, piece by piece. We're not just looking for the answers, we're going to figure out the thinking behind them. Let's get into it.
Okay, so here's our game plan. First, we'll set the scene for the whole investigation. Then, we'll nail down a really key definition, get our hands dirty with some actual cell counting, and then, brace yourselves, we'll crunch the numbers. And to wrap it all up, we're going to put on our scientist hats and critique the method itself. Ready? Let's go.
First up, let's get the context. Every good investigation needs a bit of a story, right? So picture this. A scientist is looking at a polluted pond and finds a new type of blue algae. Now, here's the important clue. This new color is caused by a mutation. That word mutation is a big deal, so you're going to want to file that away for later. So, what tools are we working with? Well, on the left, you can see this special slide. It's designed to hold a really precise volume of water, just 0.1 millimeters deep. And on the right, that's our view through the microscope. See that grid? It's not just for decoration. It is a precision tool for accurately counting these tiny little algal cells. Okay, so the exam starts us off with a pretty classic one-mark question. It's basically a little check to see if you were paying attention to that intro. And it's all about that key term we just mentioned, mutation. So here it is, question 7.1. What is a mutation? This is a really fundamental question in genetics. Before I flash up the answers, just take a second. How would you define it? What's the first thing that comes to mind? And check this out. These are all the possible correct answers, straight from the official AQA mark scheme. Do you see the pattern here? Whether you say it's a change in the DNA, a gene, an allele, or even the number of chromosomes, the core idea is always some kind of change to the genetic material. Any one of these gets you the mark.
Right, on to the next part, question 7.2. Now, this one's a little different. It's not about remembering facts from a textbook. This is a test of precision. It's about whether you can follow a very specific set of instructions exactly as they're written. So the question gives us these super strict rules for counting. You count every cell that's completely inside the square and any cell that's touching the left or the bottom lines. But, and this is the key part, you have to totally ignore any cells that are touching the top or the right lines. Now, this might seem a bit random, but it's a standard scientific trick. It's designed to make sure you never count the same cell twice, making the final count consistent and accurate.
And here's what you get when you apply that rule perfectly. Counting the ones inside, plus the ones on the left and bottom, and ignoring the top and right, you get one, and only one, possible answer from the mark scheme, 16.
All right, deep breaths. This is the big one. Question 7.3 is a 6-mark calculation, and it pulls everything we've done so far together. For a question like this, showing you're working isn't just a good idea, it's absolutely essential if you want to get all the marks.
Okay, so the problem throws a new number at us. Let's say we did a second count, and this time we only got 14 cells. Our mission is to use this number from the diluted sample to work backwards and figure out how many cells there were in 1 cubic millimeter of the original, undiluted pond water. Now, a big calculation like this can look pretty intimidating, I know, but the best way to tackle it is to just break it down into smaller, manageable steps. We're going to use this 4-step method, which is exactly what the examiner will be looking for in your working out.
First things first, we need to know the volume of the little bit of water we're actually looking at. We get this by multiplying the length and width of the grid, which are both 0.2 millimeters, by the depth of the water, which is 0.1 millimeters. That gives us a tiny volume of 0.004 cubic millimeters. Okay, for step 2, we take our new cell count, which was 14, and we divide it by that tiny volume we just calculated. This tells us the concentration in our diluted sample, and the answer is 3,500 cells per cubic millimeter. Now for a really key step. We need to figure out how much the sample was diluted. The method said that 2.5 cubic centimeters of pond water was topped up to a final volume of 10. So to find the dilution factor, we just divide 10 by 2.5, which gives us 4. This means our sample is 4 times less concentrated than the original stuff from the pond. And for the final step, we just have to reverse that dilution. We take our diluted concentration of 3,500 and we multiply it by our dilution factor of 4.
and this will give us the true concentration back in the original pond water. And there we have it. The final answer is 14,000 cells per cubic millimeter. For those of you who like to use standard form, that's 1.4 times 10 to the power of 4. Follow those steps, show your work, and that's a full 6 marks in the bag. Not so bad when you break it down, right?
All right, we've done the counting, we've done the math, but we're not quite finished. The last 2 questions test something a bit different, our understanding of why the experiment was done this way. It's time to put on our critical thinking hats.
Question 7.4 asks us for the scientific reason behind one of the key steps. So why would the scientist even bother diluting the pond water in the first place? Why not just use it straight from the pond?
The answer from the mark scheme is actually really simple and practical: to make it easier to count. I mean, think about it. If the sample was too concentrated, the algal cells would all be overlapping and clumped together. It would be impossible to get an accurate count. Diluting the sample just, well, it spreads them out so you can see what you're doing.
And for our final question, it's a classic what-if scenario. What if the scientist made a mistake? What if they used a thin cover slip that got pulled down by the water? How would that one little error completely throw off the final result? Now, this is a 2-mark question, which means we need to make 2 linked points. First, the cause. A thin, pulled-down cover slip reduces the depth of the water, which means the volume you're counting is actually smaller than you think it is. That's your first mark. And second, the effect. If you have a smaller volume, it's naturally going to contain fewer cells, so your final count would be wrong. It would be too low. It's a perfect chain of cause and effect.
And that's it. We've deconstructed question 7. But the real takeaway here isn't the number 14,000. It's the method. Follow the procedure precisely, break down big problems into small steps, and always, always question the why. So the question to leave you with is, where else in science, or even in your everyday life, can this systematic way of thinking help you find the right answer?